In this third part of our discussion on fast isosurface extraction, let's talk about an approach called contour propagation. In the previous discussion, we looked at algorithms that used a cell search approach either in the domain or in the range. The approach that we are going to talk about today closely follows from the observation that the isosurfaces are connected. Let's begin our discussion on contour propagation. The key observation is that an isosurface consists of connected components. I'm going to illustrate this using an example in 2D. So we have a 2D scalar function and we talk about iso contours that are one-dimensional. So here is a two-dimensional domain, a section of which is shown here with multiple triangles A, B, C, D, E. And we have a scalar function that is not explicitly shown, but what you see is a section of the isocontour for this function that passes through some of the triangles. So let's talk about how we would extract such an isocontour. If we are given one cell, let's say A, the shaded cell, that contains the isocontour. So given A, I know that the isocontour would traverse through some of the neighbors of A. In other words, cell A will share an iso point with its adjacent cell. An iso point is a point that lies in the isocontour. So specifically, we would like to identify the two iso points that lie on the edges of A. These are edges shared with neighboring triangles B and C, and hence we know that the isocontour passes through the triangles B and C. The idea is to identify both triangles and propagate. So this propagation, as we can see, can be computed using a very simple breadth first search. Essentially, if you think of the current node in the breadth first search tree as A, we are looking at the neighbors B and C and continuing the traversal. Of course, in the 2D case, this is a very simple path traversal. In the case of 3D scalar functions, where we have an isosurface, we would necessarily have to use the general breadth first search traversal. So if an oracle gave me the set of all seed cells such as A for each component, then I can apply the above algorithm to extract the complete isocontour. So essentially we would need the seed cells corresponding to each isocontour component. The question now is, what is the minimum number of seed cells required for the algorithm to work correctly? Compare this minimum number with the number of isocontour components. A short thought exercise for you. So here the blue edges correspond to the seed cell. You can think of one of the triangles adjacent to this blue edge as the seed triangle, and the isocontour extraction algorithm proceeds from that seed, continues till it extracts all the isocontour components. So what you see initially was the traversal beginning from the seed cells. So here it starts again. So we see the traversal. So 3 components, and then there is a 4th component which happens to be large. And later, once the isocontour components are all computed, the animation shows different isocontours which have the same number of components. So these are other isocontours. So as you can see, there are a whole bunch of isocontours for different iso values that can be computed from the same set of seed cells, namely the ones that are shown here in this frame. Of course, these seed cells are not sufficient. So later, for example, after certain events like this, we see other seed cells are required. So here are the challenges for this algorithm. We need to find the seed cells. This appears to be as difficult as the original problem of identifying the isosurface cells, because essentially we need to know all the connected components. So this may require us to search through all the cells. Of course, this is not an option. So somehow we need to maintain a small set of seed cells, one that works for all iso values. The question is this even possible? For a long time, this was not considered possible, so this contour propagation approach was not even considered. Until there was a series of papers that established how to identify these seed cells. So let's talk about this sub-problem now, identifying the seed cells. Let's take a 2D example. Here is one with a 2D domain and some isocontours shown for this scalar function defined on this 2D domain. The value of the scalar function at the vertices are shown here already. The first step towards identifying or computing the seed cells is to locate and characterize the critical points. So we understand what a critical point is. These are the points with where the gradient goes to 0. We have names for them. So for example, what we see here, for example, 5 is a local minimum. And we can see that easily because we have a linear function within each triangle. We can see by looking at all the triangles that contain 5, we see that 5 is the smallest value among, for example, 5 is less than 12, 10, 11, 20, 8, and 19. So 5 is a local minimum. Similarly, 19 is a local maximum. And as we can see by looking at all the neighbors, what is interesting is the saddle point 15. So 15 is a saddle, and if you look at the neighbors of 15, some of them are higher and others are lower. And as you walk along the ring surrounding 15, you will see that the higher values alternate with the lower ones. So you see 16, which is higher, followed by 8, which is lower, and then again high. 19 continues to lower 13, followed by high again. So this is a characteristic of the neighborhood of a saddle point, a third type of criticality. So recognizing these critical points is quite easy. One can do that by studying the neighbors of a vertex. So it so happens that the isocontour behaves interestingly at the neighborhood of these critical points. So it's easy again to see the behavior near the minima and the maxima. So let's look at the minimum, which is 5. So what happens when we look at an isocontour which is just above 5? This is going to have a single component that looks like a ring around 5. So for an iso value which is just below 5, there is no isocontour in the neighborhood of this vertex with scalar value 5. So something interesting happens at 5. A new isocontour component is born. The same thing happens at 4. 4 is also a local minimum. It happens to be on the boundary, that's just incidental. But a new component of the isocontour is born when you are just above 4. So this happens to be something like that. There is one more component, right? One more edge. Right. Now, is quite the opposite. So for example, 19 is a local maximum. So if you look at a value above 19, then there is no isocontour component in the neighborhood of this vertex with scalar value 19. However, if we look at a scalar value which is slightly smaller than 19, then the isocontour would look like a ring in the neighborhood of 19. So in this case, as we go from a lower value than 19 to a greater value, this isocontour component vanishes or it disappears. So already we can make this observation that at critical points, we have a change in the number of components of the isocontour. Now, there are the third type of criticality. Let's look at 8. So what happens so we already saw two components in the neighborhood of 8 by virtue of the components that started from 4 and 5. And these components inch towards 8. At 8, these two components merge, as is shown here. The two components that started from 4 and 5 respectively merge at 8. And after 8, they again seem to split into 2 more. Right? So this is what happens after they cross 8. So again, something significant seems to be happening to the isocontour component after it crosses 8. Now locally we cannot decide if there was a merge or a split, but since we can look at the entire isocontour, we know that the two isocontour components that started from 4 and 5, they merged at 8, and after 8, the two components indeed continued as a single component. So if you trace this entire component in the other direction, you will see that it's a single component. So it seems like the isocontour components merged into a single component at 8. Now this change in the number of connected components can be succinctly represented in the form of a tree. and this tree is called the interesting and widely studied data structure. So what you see on the right here is the contour tree. And the way to study the contour, to read the contour tree is, in this case, it is drawn in a very interesting way where the vertical direction corresponds to the direction of increasing function values. And like I said, as we increase function value, let's say from negative infinity towards positive infinity, as I cross 4, a new component of the isocontour is born, and that is tracked using this edge here between, that begins with from 4. And as we cross 5, a new edge starts coming out from a vertex called 4, 5. And these two edges merge at 8 and then continue upwards and so on. At 10, this splits into 2 and so on. And at 19, for example, there was one edge which dies. So it dies here. So the contour tree is what is shown here on the right. It is essentially representing the evolution of the connected components of these isocontours. So just to summarize, the contour tree is a succinct representation of all isocontour components, specifically their topology. It can be constructed by mapping every isocontour component to a point, and these collection of points can be recognized as a tree. The nodes of this tree correspond to vertices of the mesh. Specifically, the degree 1 nodes are the minima and maxima, and the degree 3 nodes are the saddle points. For the cases when the domain of the function is simple, like for example, if it is R2 or the sphere, then one can prove that this graph in general has no cycles and it is indeed a tree. Now, what is the connection to seed cells? So if you observe from the definition of the contour tree, every connected component corresponds to an arc in the tree. So by storing the arcs, we can actually store the set of all seed cells required to extract all iso contours or isosurfaces. So what we need is one seed cell that corresponds to every arc in the contour tree. So this is the idea behind the seed cell extraction algorithm. So here is an illustration for another contour tree. Let me explain the seed cell algorithm using this example. So here is a domain and the height function defined over the domain f. So the height function as in the function increases in the vertically upward direction. What you see on the right is the corresponding contour tree. So let's say that I would like to extract at some value here. So I would like to extract this f inverse of let me call it A, since I have C's here. So I am given A, a scalar value, and I would like to extract the connected isocontour with iso value A. Now, by looking at the tree here, I can see that it has two connected components. But the way I can recognize this is by computing the tree in some pre-processing step and searching through the edges of the tree now, rather than searching through all cells of the domain. Now when I search through the edges of this tree, I recognize those edges that span this scalar value a. And for each of those edges, so in this case there are two edges, one is C4, C5, and then there is other is C3, C5. For these two edges of the contour tree, I look at the seed cell that is stored corresponding to it, and from that seed cell I will be able to extract the isocontour components here. So each seed cell will help me extract one of these isocontour components. So this is how the contour tree helps extract the isocontour efficiently. So with that, I will stop our discussion on contour propagation. There is much more in terms of the implementation in order to realize this algorithm using the contour tree. The references that I have given here could be considered as additional reading. I'm not going to cover this topic in detail here. If you are interested, I will encourage you to read those references.